Cool.
All right. Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the three NIH-funded National Electron Microscopy Centers. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. I'm joined today by my colleagues from NCCAT, Ed Eng and Alina Kopolov, along with Michael Schmidt from the Stanford SLAC Cryo-EM Center, S2C2, and Kirsten Wheeler from the Pacific Northwest Cryo-EM Center, PNCC. NCCAT is hosting today's speaker, Bradley Guilliams, who's joining us from Colorado State. And Alina will introduce Bradley after a short update from each of the centers on the resources we make available at no cost to the research community. This webinar is a regular event we hold the last Thursday of each month, highlighting our center users who generously share the tips and tricks that have enabled them to make use of cryo-EM in their research with the help of National Center resources. So you can save the date now for October talk with Rocky Rajan and Chandusi Ganjuli from the University of Oklahoma, who will talk about visualizing the DNA cleavage fidelity of CRISPR-Cas proteins using cryo-EM. So if you want to find out how spooky CRISPR-Cas systems are on Halloween, please come join us next month. The registration link is already available on the events page at cryoemcenters.org. All right, and finally, before we kick off today, please use the Q&A box to send questions at any time during the talk. I'll moderate questions at the end of the talk, but feel free to send them at any point, and we'll get to those at the end. So now I'll turn it over to the centers to let us know what's new. And I will turn over first to Kirsten.
Sorry about that. Mute button didn't want to go off. My name is Kirsten Wheeler, and I'm the administrative coordinator for Pacific Northwest Cryo-EM Center. PNCC has two types of proposals. First is our screening access proposal, which allows one user to come on site for up to 5 days of sample preparation and screening with staff. This proposal is aimed at low resource institutions. The second proposal type is the general access, which awards up to 480 hours of microscope time a year for 2 years. This time can be used for screening or data collection. Approved general access proposals are also eligible for our training workshops, which cover topics from sample preparation through data processing. PNCC has 6 microscopes. We have an Arctica with a K3 camera for screening and 4 Krios instruments for data collection equipped with K3 and Falcon 4i cameras. 3 of our Krios are equipped with Katon energy filters, while the other 2 have Thermo Fisher energy filters. Our Krios 1 just completed its upgrade for a Selectress X. The newest member of our microscope family is Glacios 2, which will be utilized for both screening as well as data collection. For sample preparation, we have our Vitrobot as well as our Leica GP2. If you're interested in working with PNCC, please go to pncc.labworks.org and apply today. Deadlines for proposals are the first of each month. In addition to that as well, we also offer trainings that we have each year. If you'd like to take a look at those, go ahead and do that. One of them is open to the public, whereas the rest of them are available to our general access proposals.
All right, thank you, Kirsten. And next I will turn over to Mike to let us know what's happening at S2C2.
Hi, I'm Mike Schmidt, and I'm one of the co-directors of the Stanford SLAC Cryo-EM Center. In addition to the 4 Krios we have, we're getting a Glacios and a Tundra. We have all the specimen preparation equipment you might desire, and we offer, of course, service. Sample preparation and optimization and training. Our training was really updated this last week. We had a CryoSpark workshop with Ali Panjani, the developer of CryoSpark, and it went through all the intricacies of this CryoSpark workflow. It was amazing. It was amazing workshop. We had 200 Zoom attendees and 80 on-site people. It was great, and it'll show up in our workshop archive on our site. Check it out. It was really great workshop. And this week we are hosting SSRL at Slack is hosting the SSRL user meeting. We had one session on cryo-EM to tell all the X-ray crystallographers and other people that use the synchrotron and the LCLS, the laser, how to about cryo-EM and how it can help them in their research. So we're a one-stop shop for all kinds of structural biology. So just check us out.
Thank you, Mike. And finally, I'll turn over to Ed.
Hi, I'm Ed, the manager of NCCAT here in New York. We have 5 Krios instruments, 2 Glacios instruments, a Tundra, and a whole slew of other instrumentation for not only data collection, grid preparation and screening, and cross-training. Something I would like to highlight, in a couple of weeks, we'll have our Integrative Structural Biology Symposium. On our workshop Wednesday, we are oversubscribed. However, we are now offering a second offering of our PyMol 3 workshop. That'll be led by Tom Stewart, a senior developer and product manager of PyMol at Schrodinger. If you're interested, please message me, or you can go to nccat.nysbc.org to find out more. Thank you.
Thank you, Ed. And I will stop sharing and let Alina introduce today's speaker.
Hi, everyone. My name is Alina. I work at NCCAT, and it's my great honor today to introduce Bradley Guilliams. He's one of the NCCAT's long-term users. He first applied to us with a sample preparation and screening axis and very quickly graduated to Krios axis. We love him very much. Bradley is a PhD candidate at Colorado State University, Akerson's research lab. Outside of the lab, he has a very interesting life. He does rock climbing. In fact, this summer, him and his friend build a big rock climbing wall in his garage. He loves cooking with his wife and having friends over. So if you're ever in need of a good recipe, please reach out to him. We'll share his email with you. Yeah, thank you. Bradley, please take us away.
Awesome. Thanks for that. No, it's been great working with everybody at NCCAT and over the last I guess almost a year at this point, where I first reached out. So yeah, thanks for having me. Today I'm going to be talking about some of the research going on in our lab. Let's hide this at the top here. There we go. Okay. So today I'm going to be talking about some of the research going on in the Akerson Research Lab, sort of setting up some of the motivation for the single particle cryo-EM project that I've been working on and I'm still actively working on. So it'll be sort of going to show you the result of a lot of work that's happened in the lab leading up to this, where we're developing a clonable contrast tool for use in electron microscopy imaging, as well as some fluorescence imaging. And then after that, I'll transition into the single particle cryo-EM project I've been working on that is still, again, actively happening. So yeah, let's get started. So to set the stage here, I first want to thank, again, all of these wonderful people. This is from our lab retreat we had back in the summer, where it's current and alumni, we all come together, try to about once a year. And of course, lots of collaborators at all of these different institutions and organizations throughout my time here. So without any of them, this would not be possible. So to, again, set the stage here, we're talking about clonable contrast tools. And so we think about contrast in the electron microscope. If we have some electron source, some biological species of interest, a protein or a cell, is going to interact comparatively weakly with the electron beam, whereas something that is more electron dense, like an electron, like a nanoparticle, is going to interact much more strongly with the electron beam, scatter the electrons, giving rise to contrast. So these things I'll call tags, right? There are various forms. They add contrast to biological material, and they allow us to visualize proteins by proxy. So we see the tag, and because the tag is proximal to our protein of interest or what we want to see, we are able to then see that protein of interest, right? And I've shown here an example of like an antibody that's got an electron dense particle on the end of it. And with certain tags, you end up with this, distance between your actual electron density and the thing you want to see. Right, so this is something to keep in mind. And we're talking about clonable tags. So these are analogous to, say, green fluorescent protein, but there aren't many tools that are analogous to that are available to electron microscopists. So our lab has defined this paradigm of the clonable nanoparticle. This is our proposed solution, I guess, or one proposed solution to the grand challenge in biology that is, contrast. With this paradigm, we have a nanoparticle whose growth and synthesis and the properties are defined by some protein or protein peptide construct. And of course, this protein construct, goes back to the DNA. We can clone and have control of the DNA, and therefore we can control the properties of the nanoparticle in different systems that we put this protein into. All right, and so a generalized application of this would be, say we want a clonable cobalt nanoparticle of defined size. Again, it's got this, it's made of cobalt and it's some icosahedral structure, right? We would use a clonable nanoparticle protein that controls the synthesis of this particle inside of cells, right? I show these inorganic elements, which are essential for life, because many people think of metals in biology and they start to think a lot about toxicity and things like that. But the reality is there are a number of inorganic elements that are essential for life, and they're really not all that to be, they're not, shouldn't be that scared of them, I guess. So we can think about some of what I'm saying, and going forward, think about things in the context of a checklist for what we want in a cryo-electron microscopy tag, right? So we want something that is clonable or synthesized and functions intracellularly, in, I guess, much the same way that green fluorescent protein you express inside a cell, and it goes and attaches to, it's attached to what you want to see. We want this to be able to function a variety of biological systems. Sort of straightforward there. We want our tag to be small and provide proximal labeling. So we want small in that we don't want some electron dense tag to obscure the thing we're trying to see. So what we're saying, if proteins that we think are on the order of 10 nanometers or more, right? We want a tag that in its entirety is something less than 10 nanometers, give or take. We want some stoichiometric labeling, so we should have a single electron dense tag per protein of interest. We don't want a background in the cell that is from our clonable tag. And these, of course, should be identifiable in electron micrographs. Sort of straightforward there. And I've got this bonus point here for identifiable in fluorescence micrographs, right? This would be to facilitate something like correlative light electron microscopy. This is an area that we're working pretty heavily in right now. And so I'll set the stage for that very briefly before continuing. So some motivation for why we want labels for cryo-clem, right? If we think about the idealized view of the cell, right, we have all of this structural information. Here we're seeing larger macromolecules, and they're colored different colors by their identification, by their molecular identity, right? So we have all of this. The reality is as well that all of these empty spaces are filled with smaller proteins that we can't see directly, right? And when we actually look at it in electron micrograph, yes, we see and retain this structural information, but we lose a lot of this this chemical or this like identity the identity of all of the things we're trying to see, right? But we can, I guess, make out some of these structures, right, based on their morphology. You have these long stringy things versus blobby things versus these things on the edge of the membrane. So, okay, but say you want to see something that you can't visualize directly, right? Maybe we use a fluorescence tag to give us, into the neighborhood here. But of course, with a fluorescence image, we're given, the identity because we know that clonable fluorescence tags are attached to something that you attached it to your protein of interest, right? But in our fluorescence image, we lose a lot of the structural information. And when we then have this area where to look, we can zoom in with our electron microscope. And if we've got an electron dense tag, we can see, what were our protein of interest at this higher resolution by proxy of these electron dense tags. Okay, so this is the area where our lab is sort of working. We're developing tools for cryo-EM and cryo-CLIM. Okay, so I've told you about a very generalized clonable nanoparticle. I'll tell you about our clonable nanoparticle. So this is based on a protein called glutathione reductase-like metalloid reductase. It's a mouthful that I will hopefully never repeat again. Right, it's GRILMR. Again, we made this acronym before we were thinking too far ahead, and now we're stuck with GRILMR, which sort of sounds like a strange noise you're making with your mouth. But this is our protein, and we're stuck with it, right? So we isolated a protein from a microorganism shown here with these, red streaks on agar that's been supplemented with selenium. So it was living in the roots of Princess Plume, which is a selenium hyperaccumulator. And it was the single protein takes and makes selenium nanoparticles as a way of increasing its tolerance to otherwise toxic amounts of selenium. Right, this is some tomography of the microorganism that we pull this out of, and it makes these large selenium nanoparticles as well as smaller ones. So this polydispersed size here, right? We can clone this into E. coli, and it affords a similar selenite tolerance at normally toxic levels. Okay, so thinking about what we know about what we want for a tag, right? This native enzyme, GRILMR, it makes particles with a very large dispersity in sizes. These large particles are not going to be super useful because they'll obscure anything we want to see. But the smaller particles, yeah, we want, kind of the smaller size that's still going to be plenty electron dense for easy identification in electron micrograph. These particles were prone to aggregation, partially due to them not being retained at the enzyme because it's not really anything holding them there necessarily. But by adding a selenium binding peptide that we isolated from a phage display library, attaching these two monomer units, this is an obligate homodimer, and all we've done is attach the two chains together. We added this selenium binding peptide, and with just these two small modifications, we control the particle dispersity to where in cells we're making particles about the size of 5 nanometers, plus or minus about a nanometer. It reduced the aggregation of particles, and of course they're also retained at the enzyme as well. And as a bonus, it actually sped the enzyme up. So cool. So using this as a tag, right, we can create some generic chimera here with a protein of interest. Through transcription and translation, we end up with this covalently linked protein of interest and clonable selenium nanoparticle protein. Well, we add selenite and NADPH. NADPH, of course, already in the cell. We don't have to add this to cells, but this reaction happens outside of cells as well. We end up with this. Clonable selenium nanoparticle retained at the clonable selenium nanoparticle enzyme attached and proximal to a protein of interest. All right, so this is a generalized view of this, where we tested this using in E. coli using FTSE, which is a filamenting protein analogous to tubulin. And it has, importantly, known localization. So when cells aren't dividing, they're sort of FTSE is at the towards the membrane, the outer membrane of E. coli, and during cell division, it's implicated in initiating the Z ring that helps cells divide. So again, we have known localization, and we we overexpressed this construct with FTSE monomers and our clonable selenium nanoparticle. And upon polymerization of FTSE, in combination with genomic FTSE, we end up with these FTSE filaments that are dotted with selenium nanoparticles. Shown here on the right two panels, C and D, are examples where cells had actually burst and these FTSE had come out, these filaments came out, and they're sort of dotted with selenium nanoparticles. On the right is just a segmented version of this here. Okay, so this is all still in vitro, right? We look inside of cells. These are high pressure frozen, freeze substituted cells. We can easily pick out these electron dense particles. This is a handful of slices. They can be segmented automatically with a simple pixel thresholding, so supporting the idea that they are easily identifiable in electron micrographs, right? And shown here is just tomographic reconstruction of the cell, as well as showing that most of these particles are proximal to the membrane, right? We have additional data at a cell division point showing selenium nanoparticles along these FTSE filaments. But there's not enough. I'm happy to share that data after the fact here, right? So I've said these are high pressure frozen freeze substituted. Right now, I've been working for a long time, longer than I'd like to admit, on trying to get this working in cryo-electron microscopy, or in cryo-electron tomography rather. Because these are E. coli, even though they are quite small, they still need to be milled, which has served as a fairly big bottleneck for us. And even with milled cells, E. coli are quite dense. And so this is sort of the wild type enzyme, Guilliams. So it's making these really large particles that we can see. But when we mill and look at sections here, I've picked out a handful of particles throughout this, but these were all manually picked and with some difficulty. I'd say that this is an area that, again, I'm still actively working, and we have hypotheses about why it's been giving so much trouble, again, for another time. Right, so if we return to our checklist here, right, I've shown that this is a clonable, that if we think about this for a clonable selenium nanoparticle, it is clonable, as the name suggests. It is synthesized and functions intracellularly. In fact, we pulled it out of a microorganism that it was already working in. I have this sort of orange dashed line for functions in a variety of biological systems, right? I've only shown you data for E. coli and by proximity, another bacteria, right? We have a collaborator that is working in Drosophila, and there's some promising data there, but I don't have any sort of definitive imaging data to go along with that. So it's sort of to be determined, and we're also getting things going in a couple of other organisms, work done by other people in the lab, right? This is small. These nanoparticles are 5 nanometers, and if everything's working right, the label is proximal, right? The distance between our particle and our protein of interest is going to be significantly less than 10 nanometers, or very, very worst case, about 10 nanometers from like the center mass of our protein to the center mass of the protein of interest, right? They're identifiable in electron micrographs. Okay, I haven't said anything about fluorescence thus far, but with a relatively simple reaction, we can add a transition metal cation such as zinc, and it converts this otherwise amorphous selenium particle into a crystalline quantum dot. The intricacies of this are still being worked out, and much of the work in cells is going to be done by another graduate student who's just starting her PhD by the name of Taylor. So watch out for what she's doing, right? We did show, though, that this does work in theory. So with a very crude correlative light electron microscopy experiment, I say crude in that we didn't use any, we manually overlaid these images, right? So these are E. coli cells expressing the clonable selenium nanoparticle, where we've made zinc quantum dots, zinc selenide quantum dots, and they are localized at the pinch points of cells as anticipated and at the poles, again, as anticipated. So, okay, I've told you a lot about electron tomography. This is a single particle cryo-EM webinar, right? So why am I doing single particle cryo-EM when we've got something that's working? Okay, we have bigger goals here with green fluorescent protein, right? There's a whole toolbox of variants that express different colors, right? So you can do simultaneous localization of multiple species of interest. You can look at how these things are interacting dynamically, which is, of course, a strength of fluorescence, but it's enabled by these tools, right? So right now, I've told you about a single clonable nanoparticle, right? We can use this, and we have shown that it's working. We have a working system, all right? So, but how can an enzyme do this, right? There's no structures of any proteins that are, I would say, very closely related to this. So it's sort of this unique enzyme. We have no structure of it. So how does an enzyme make nanoparticles? And extending this, right? Can we leverage this to make different clonable nanoparticles that are elementally distinct or visually distinct? Of course, like, something that's a triangle is going to look different from something that's a sphere in the electron microscope, right? So this is sort of the motivation. We want to understand how things are making, how this enzyme is making selenium nanoparticles. All right, so shown here are AlphaFold models of our protein. And the experiment we're setting up here to try and understand this is, of course, we need a structure of our protein of Grillmer. This is a 100 kilodalton homodimer. It doesn't really exist as a monomer. We don't really see, we do any kind of purification, we don't really see it exist as a monomer. So very tight interaction in this homodimer form, right? We also want to see where selenium binds within this protein, as well as NADPH, which we know is implicated in the actual chemistry happening to make nanoparticles, right? So our goals here, again, determine a near atomic structure of Grillmer, and to determine that structure with selenium and NADPH within it. Okay, cool, right? So finally, we're doing single particle cryo-EM. I'm going to be walking through this workflow here. So you'll see this on a lot of the slides coming up as I'm doing sample screening, sample prep. And then we'll move on to some data collection, data processing, which will have a different workflow. The goal with sample preparation and screening is to have grids that are Krios ready. Okay, so we do our screening on something like a Glacios or similar lower KV scope, and then do our long automated data collections on the really powerful microscopes. So I applied to NCCAT, their screening program. We don't have cryo-EM resources at our institution. Rather, we don't have a cryo-electron microscope. Our lab, as of recently, has a plunge freezer, but again, there's no microscope to screen this by. So did some negative staining of our purified protein on our gel microscope, right? Things looked okay. We had some biochemistry showing that our protein is purified, but this is pretty trivial for us because this is a protein that we've been working with for a long time, and we use lots of we use our purified protein in lots of different assays, right? So there's not really interesting story there for me to tell, right? I arrived at NCCAT, and they had this cool thing called a mass photometer, and I was like, well, I got to try that. So this is a way of assessing protein purity. Uses light scattering, hits protein, and the way that light interacts, you get a peak that corresponds to the weight of your protein. So sort of unexpected or expectedly, we have this peak here corresponding to our purified Gromer homodimer. But somewhat unexpectedly is we had this not insignificant peak corresponding to 2 of these homodimers coming together. Here, the counts are about 22%. So I'm not trying to say that there is this other species we need to be worried about. I guess we'll, think about it later or see if it, crops up later, right? But ultimately, pretty happy with our purified protein here. So thinking about freezing conditions, then I was working with either gold or copper 300 mesh quantifoils, standard grids here, and had a variety of blot times working on the Leica EMGP. So most people are doing single particle, probably using the VitraBot. We're using the Leica counterpart, largely because we knew we were going to be getting one in our lab. It's one that I had used before for cell stuff. And so it was sort of, if I was going to be making samples at home, I was going to be making it on this. So better to optimize it on something that we're going to be able to use in the future, right? So on day 1, the lower times here gave us thicker ice. You can see, can't really see much of the grid squares, right? On some of these longer blot times, we had this kind of gradient of ice with some regions where the ice was relatively thin. Our second day, the times changed to shift slightly longer. And we also updated the blot position on the Leica EMGP. So the VitraBot, as I understand, has a blot force as a way of how hard you're wicking, right? But the Leica EMGP does not have something like have a measure of this, and it only has a single arm that it uses to blot proteins, right? But you can manually move the position of the arm as a way of sort of getting a more firm blot. In day 1, I was getting sort of inconsistent blotting. And by updating the blot position and focusing on these longer times, I'm getting consistently things in this 4 to 5 second range that look okay. Here is a slightly thicker 4-second blot grid, but there were still regions of this grid that looked thin and usable. Okay, so we've narrowed down kind of the blot time on some known grids here. Now we've got, of course, our protein. So day 1, I scanned a number of different concentrations on the grid, and it was really only this lowest concentration where we didn't see too many aggregates. I would say this is an okay grid, right? There are some clumps here, some contamination, but there is, individual particles, right? Day 2, we kind of had a narrower range of concentrations in this 0.75 to 0.5 mg per mil of protein on the grid was sort of the sweet spot. There were still some aggregates, but going much lower than this, and we had lots of empty space on our grids. So lots of stuff that just, there's no particles. So, okay, in the following days, I sort of made some replicates of these kind of grids that I knew were pretty good. There's, of course, variation in when you make grids. And so making a bunch of replicates and screening those kind of gives you some different options, right? Our presenters note here on day 3, we sort of tried gold foil grids sort of as a curiosity, wonder what will happen. And at this point, I already have conditions that I feel good about for carbon film grids, which of course don't translate to gold foil, which sort of makes sense, right? I guess at the time it was, okay, yeah, that confirms maybe some hypotheses, but ultimately, I didn't have a single image with a protein in it through like 6 grids or something like that. So I guess ultimately the grid type you use, you want to stick with that, or you have to sort of optimize the freezing conditions with the grid different grid types. Okay, so towards the end of throughout the week here, I had several grids that were okay, and we wanted to do this small data collection. You do some small data collections. Let's say one, if you're new like I was, it was hands-on time practicing using something like CryoSpark, which is what I've used for all of my data processing so far. So it's hands-on time with somebody like Eugene Chua around to help you, and Kasahun to help you figure out how to, which button to click, right? But it also gives you an opportunity to look and see if there are preferred orientation or otherwise challenges with your actual protein. So different from how it's distributing across the grid and different from how thick the ice is, right? So we got a small data collection of 500 micrographs, worked it up in an afternoon, and lo and behold, I told that my structure is 4.85 angstroms. I'm told, don't trust this. At the time, I'm thinking, oh yeah, it's maybe like a little more than 5, but yeah, holy crap, this is, this is cool, right? This is a glacios, 500 images. We collect 10,000 on a Krios, and this is going to go down to, 0.5 angstrom, no problem. So the last thing, right, we've selected, I've got now all these grids that we've screened. I want to select Krios ready grids in some priority list. And here you're sort of looking at all of these different magnifications. I'd say the most important ones, at least for me, were looking at the atlas, so looking at ice thickness across the grid. A gradient is good because even if you're collecting, it gives you different kinds of ice to collect in. You want to look at the hole level images, right? You want sample to be in your holes. If your ice is too thin, then you start to get lots of holes that are empty and there's nothing in them. And when you have a long automated data collection, you collect a lot of nothing. And then, of course, you look at the protein level images to make sure that there's protein there that looks good. Okay, so after this, I applied for the RAP1 access at NCCAT. Ultimately, came back to screen some more samples for these second and third experiments.
Oh, Bradley, I think we lost your sound. I don't hear you.
No?
Now it's okay.
Yeah, it sounds like your headset died.
All right.
Oh.
What's going on here? Can you hear me all right?
Yeah,
Yes.
I hear you now.
Okay.
Sound quality's still good.
Okay. So depending on where I cut off, I applied for Krios access at NCCAT. Ended up having 3 separate data collection sessions. The screening of these latter 2 samples was pretty similar to the first one here. I sort of had a starting point considering the protein sample is the same. So cool. So I've been working on this first data set for now about 2 months. I had some trouble getting access to computational resources. Our lab workstation never really came alive. And so I've now been using computation resources at Chan Zuckerberg Imaging Institute, where we are collaborating. But so we have this data set of Gromer with about 10,000 micrographs collected on a system with a Gatan K3. So we've just talked about sample preparation. We've now had Krios data collection, right? We're going to this data processing flow chart. Right, there are many different ways to think about data processing. I am by no means the authority figure. This is a way that I found that is convenient to think about it. And there's iteration both between steps as well as within each of these steps. So keep that in mind, but this is how I'll be sort of walking through what I'm doing. Okay, so I'll tell you a story about baby's first cryo-EM workflow. with Krios data. So I imported some micrographs. I did some blob picking. I got some classes with some trouble. I'm turning knobs and dials that I don't really know. But eventually I get some classes that look okay, maybe. I take those blob picks. I've learned that I can use them in a template picker. The template picker is going to pick a bunch of particles. And again, with some trouble, I get classes that look okay, right? I can see some secondary structure in these classes, right? Keep in mind, I'm a chemist, right? I don't know any better. This is my first cryo-EM workflow. Okay, so I take these, I get an initial structure, I do a homogeneous refinement, and wow, I'm at 3.5 angstrom. Cryo-EM is easy. This is great. I'm having a great time. Then I can't get below 3.5 angstrom. Okay, that's fine, right? Maybe 3.5 angstrom is okay for right now. I just kind of, I'm sort of blindly pushing ahead here. I just kind of want to see, the end goal here. So I was at the 3-dimensional electron microscopy GRC this summer. Daisuke Kihara gave a great talk. We had lunch after and invited me to use these computational resources that he hosts on his lab website and server. So again, these are very easy to use. You submit a job to use one of their tools. You upload some files. For somebody that, again, has never done structural biology of any kind, like these were very intuitive, very nice tutorials, very easy to use. I encourage you to check these out. They have a variety of tools, largely in this model refinement, model building kind of area. Right. So I use one of their deep learning tools, and I see some pretty bad steric clashing here in the middle here, right? Maybe I can fix this. I don't know, maybe. I boot up Coot for the second time ever in my life, maybe. And right, something's not quite right here. So one of the last things I did, I reached out to a friend and collaborator of my boss, Dave Bushnell. Like, hey, is there any way to fix scaling in Coot, right? This map in blue is way smaller than these coordinates from an AlphaFold model. Something's not right. Like maybe it's just if I make it bigger, it'll fit. Okay. Something's not right though. So let's take a step back for the first time. Let's go back to the particles here. If I look at my Glacio small data collection, I look at the scale bar for these particles. Classes look, not commenting on those, right? But I compare them to the Krios data, and the scale bar is half the size. Okay, something was wrong before this. Look at my CTF, it's flat. Didn't notice that, right? I think there's supposed to be features there. And you collect data in CCAT, they have this awesome reporting and interface where you can get all this data, right? I scrolled right past the nice summary at the top. I go to the bottom, it told me I had a Gatan K3 and my pixel size is 0.4 angstroms. I inputted that, right? What I should have seen was this nice table at the top and the images that I used, which were aligned as weighted, where my pixel size was 0.8 angstroms. So, right, sort of explains everything that I was seeing with, right, some caveats, right? I did learn some things along the way, right? And I'll talk about how I'm now doing things with the correct pixel size, right? This is still ongoing. So one of the things I learned, if you read the tooltips, the circular blob picker is actually 3 blob sizes. I sort of assumed the first couple of times I was using this was like some continuum of blobs from a minimum and with a minimum and max. It was going and picking all this stuff, right? The elliptical blob is actually just this a single ellipse where you're defining the max and the min axes of this. So again, equipped with my correct pixel size, going back through these steps here, doing my coarse particle curation, But blob picking is turning up a lot of junk particles. To those of you that are experienced, this probably doesn't surprise you. And I think that the fact that this is quite a small protein with a comparatively low signal to noise ratio, I'm picking lots of junk. The blob picker, of course, picks anything that looks like the shape that you're telling it, the circle or the ellipse. So pulls up a lot of junk. With some effort, I pull out a handful of classes that look okay. The only way I was able to do this was actually by changing these parameters within the 2D classification. I used number of iterations in the batch size per class. So this I was doing before as well. I couldn't get any classes on my previous data set until I did this. I couldn't get any classes that looked like my protein. So, okay, so I'm sort of repeating this a handful of times, but I'm having a hard time getting rid of junk, and I've got a lot of junk that's kind of combined with things that look like my particle. I kind of carry through a little bit. I do some initial structure, and I'm kind of hit some wall, and what I learned this time is ask for help from people that know what they're doing. So some words from the wise. So our collaborators at Chan Zuckerberg Imaging Institute, I showed them like what I was doing. Bridget, if I had to boil down what she said, it was basically to underpick and clean things up with 3D classification, something I wasn't doing. And Reza emphasized that everything was really like the particle picking was this really important aspect that I wasn't giving it kind of the care that it needed. Okay, so equipped with this new perspective, and they also told me to stop working with my entire data set, which, I'm working with this huge cluster. They have IT support. I'm like, oh, I'll just process everything. That's fine, right? But the things, you notice some of the times, jobs are taking, 5, 6, 7, 8+ hours, right? That's not a good use of my time. So I used this particle sets tool under utilities to break one of the blob or template picker jobs where it gave me something like 6 to 20 million particles. So I broke this into batches of about 100,000, which are much easier to work with. I took these and I classified them into like 100 or 150 classes, still with these increased number of iterations and this increased batch size. I separated into a bunch of these classes. I'd throw away a handful of them that were like obviously junk to me. Right? I don't want to comment about like I didn't know what I should keep versus not, so I'm just get rid of things that are definitely junk. I repeated this 20 to 25 times, which is quite a lot with 100,000 particles probably, but it worked for me. So at the end of this, I've got about 7,000 particles with classes that look like my protein, sort of. I used these to train a Topaz particle picking model on a subset of my micrographs. So it's another utility to just break your micrographs up into different batches. So I trained this model, and I used this model. I tested it on a small data set or a small subset, and things looked like they were working out okay. So I went ahead and maybe foolishly, but I was excited. I went ahead and picked on the remaining 9,000 micrographs, did some 2D classification, and already I'm going from less than 10 of 150 or 200 classes looking good to like maybe 50, half, like a third to a half of my classes like actually looking like a protein. And it also only picked about 1 million particles across all of those micrographs. So seems like it's picking a lot less noise. So I take these, I classify them in 2D, and then I do ab initio reconstruction with multiple classes and follow with heterogeneous refinements. And I end up with 2 classes that look sort of like junk, and one that looks a little bit more like my protein. okay. And we compare this to the AlphaFold model. I know the rotations aren't quite aligned here, but you have to trust me. It is looking like this model, right? The model shouldn't be ground truth, and I'm not trying to treat it as such, but it is a reference point as I'm figuring out what I'm doing here, right? And ultimately, I'm still kind of going through this iteratively through this particle curation. So by classifying in 3D in these different ab initio classes, I can get rid of the classes that are just made of particles that are junk and keep the one that looks like my protein. Right now, you'll see there's lots of stuff floating on the outside here. And comparatively, I'm seeing more detail. I'm still very wary to trust this FSC, this resolution that it's telling me. And in fact, I have learned recently at the CryoSpark workshop that Michael was talking about in the intro. I was able to attend for about the first day of that. I learned that a shape like this tells me there's something wrong with my mask, or there's masking, or something's too tight, maybe, right? I don't know anything about masks, so that's right. I learned that last week, so after this, I'm gonna probably eat some lunch, right? And then I'm gonna look at this cool mask creation video posted by CryoSpark. I mentioned a while ago, right there, these dimer of dimers, and I should have thrown it up here, right? But I'm thinking that a lot of these second particles, the density is getting included, and it's maybe trying to align somewhere in between these two particles, as opposed to just aligning with one. So I'm going to play around with masks with a single particle. That's where I'm going next. Any other advice? Again, happy to learn from those that are more experienced, right? But I am, again, learning this as I go. We're not a lab that does single particle cryo-EM, so but I've had a lot of fun with it. So another consideration, hopefully it isn't a problem, is that my ice was relatively thick compared to my protein. So I think there's a way you can see in the FSC curve whether or not you're actually reaching that limitation, but I would say I'm keeping this in the back of my mind, but not, counting my chickens yet, right? And of course, I've still got these 2 extra data sets that I get to play with. And I think that I'll probably end up doing some interesting things with masking, maybe doing something like a local refinement around these areas where things are, we think things are bound. Right? So I'm in a couple different places with this. Very exciting. Happy with it. So to bring this all back around, right, we want to understand enzymatic nanoparticle growth using our clonable selenium nanoparticle. We've got these 3 data sets. We want to see where these small molecules are binding. And all this is moving towards building this toolbox for electron microscopists to be able to localize multiple species of interest with an electron microscope. And of course, at the beginning of the talk, I told you about our clonable selenium nanoparticle, how we're using this, and again, some active work happening with another graduate student pushing the cryo-chem labels portion. So thank you. Happy to take any questions. Yeah, thanks for your time.
Thank you, Bradley. That was amazing. You're not hearing the applause that's out in the crowd. We agree, cryo-EM is just so easy.
Yeah.
That was fantastic. So if people have questions, there's a couple questions already, but if some of you have questions and or ideas, feel free to use the Q&A to send some things in. I'll start with a question back more to the intro. What's the actual size of the selenium nanoparticle? Or if you don't know the actual size, maybe like relative size compared to
Yeah, so inside of cells,
yeah.
they're about 5 nanometers, 5 like plus or minus a nanometer. Outside of cells, and this is something that is sort of something we're trying to figure out. Well, it makes larger particles when it's kind of just in buffer. Yeah, happy to talk more about some of the hypotheses as to why that happens, but ultimately we're able to control the size inside of cells to be about 5 nanometers with this binding peptide and the amount of selenium we're using, stuff like that.
All right. Then there was a question from someone who has the experience of getting a 3.2 angstrom refinement and sort of having struggles getting that to go down. And I think this question was sent in before, Bradley, you sort of walked through some of the steps that you use to approach that problem. And I'm guessing that is one of many approaches. There's lots of things to go along. So I don't know if there's a specific way to answer that question. But I guess to know other people have had a similar experience.
Yeah.
Yeah, I guess
I was
go ahead.
like, I think that what I have gleaned thus far is I'm not limited by pixel size. Like I'm not at the Nyquist limit. I think looking at the FSC curve and that graph that spits out will tell you a lot about some of the things limiting your resolution, maybe. That's my, I guess, interpretation. I guess if it's like not falling to 0, I think I read this on like a forum post for CryoSpark that like if it's not falling to 0, you may be limited by like ice thickness. I guess is that correct from somebody that knows more than me? But yeah. Having fun. I think my problem is junk in my particle stack, and that was my problem for a while. Now I think I'm limited by the alignment to a single homodimer structure.
Yeah, there's a question from Dimitri asking if you used an energy filter.
I believe so. Yeah, all of the Cryoses at NCCAT have energy filters, and they're either 10 or 20 EV slits. So.
Yeah, and then another question. Yeah, the FSC on your slides 26, 32, and 34. Yes, is how would you explain the FSC looks challenging? And maybe
what were the other numbers? This is 34. Maybe I'll just find one.
Actually, Dimitri, I've let you talk if you want to unmute and say if there's something you see in that FSC curve. Yeah, I think slides. His microphone. He says his microphone is not good. But yeah, I think you did mention what you saw in this FSC curve. So
yeah.
Dimitri, let us know if that didn't answer your question.
Yeah, there's an earlier one where there's like bumps kind of along the way down. I'm not exactly sure what that means, but I know that's not supposed to look like that. I think it may have something to do with the mask that it's using for there's a if you look up common, like there's a CryoSpark has great documentation. One of the pages I was looking at recently was talking about interpreting GFs, the FSC curves. And I think the spherical or the mask it's using to calculate the resolution, if there's a problem with that, sometimes I think this will bounce up and down. Yeah, I'd say that I've kind of already, I don't know much more than that. I think where, like if it needs to fall to 0, and these are ultimately falling to 0. So there's not something, again, I think I read something about that being related to maybe ice thickness being a limiting factor, but again, I've reached the edge of my knowledge thus far.
And actually, quickly, Bradley, if you have a couple minutes, we'll stay and get to the last couple questions. But for people who have to pop up at 1 o'clock, thank you for joining us today. The Cryo-EM centers are here to help you support your science. Similar to Bradley, we hope you come to us with your questions and your projects. So let us know how we can help you. And we'll put the recording up that will include these last few questions at the events page at cryoemcenters.org. So Bradley, can you stay another couple minutes to finish the questions? Okay.
Yeah.
Also, Dimitri did say his opinion is if you clean your data, you'll get higher res. And good luck.
Yeah, thank you. Thank you.
All right, then there's a question about how long the linker between your protein of interest and your gromer. Do you see a shadow over the protein of interest from selenium particles in your tomography?
So how long is the linker? I'd say we're playing around with this. Something on the order of like 12 to 15 amino acids. We're with our collaborations at Chan Zuckerberg Imaging Institute. There's lots of people interested in really improving subtomo averaging. And so one of the problems they're talking about is their alignments in these subtomograms. And so, you know, right now the linker is flexible, so there's not some rigid orientation between the actual particle, the nanoparticle, and the protein of interest. But we're thinking of kind of adding a more rigid linker as a way of kind of defining this position between the nanoparticle, and the protein of interest a bit more. As for a shadow, not really. So I would, not that we've observed this being like, I guess, a limitation thus far. Selenium is in the TEM actually not that electron dense compared to something like gold. But in the TEM, you're not really taking advantage of much like C contrast. So normally or like scanning transmission electron microscopy, you know, your contrast is much more related to the atomic number of the element you're using. Whereas in TEM, as I understand it, it's much more like mass thickness, much more phase contrast contribution, contributing to your image. And I would say resultingly, because it's an amorphous particle, it's actually not that dense. We don't see much of a Fresnel fringe in like 2D images with selenium nanoparticles inside of cells, something that you might see with a kind of a large gold particle. Or we do see that if these selenium nanoparticles grow much, much larger. And so I guess some of the work with CLEM imaging, making these bimetallic nanoparticles, some of which are fluorescent, some are not. Like a copper selenide particle is not fluorescent, but because it has this crystallinity, that interacts a bit more strongly. So kind of gone on a tangent here, but I'd say not that we've noticed. And I'd say the target audience for our tag, like the most textbook target audience, is going to be proteins that you can't directly visualize. So things in the, you know, in electron tomogram. So you're not necessarily going to be worried about shadowing because you wouldn't really be able to see the protein anyways. And so you're really just looking for the tag. So.
And then another question on that topic is, how easy is it to regulate the particle size in the cell? I think you mentioned that briefly, but.
Yeah, so I think it's definitely something that would be optimized for, you know, each system. But we're starting to think a little bit more in the context of like homeostasis inside of cells, and we think that we're able to kind of clue into that a bit. Yeah, I'd say we've optimized something in E. coli, and it's pretty reproducible, I would say. And going beyond that into other organisms, it definitely would be something that has to be optimized. We think that the binding peptide does help with this. And I'd say, you know, we are trying to see, I guess, we may find that when we clone this into other organisms, as long as there's enough selenium around, and we have this binding peptide on it, that it may just kind of stop at 5 nanometers. It may be enough on its own. I mean, there's a lot of literature about peptides controlling nanoparticle synthesis. You know, there's a lot of examples with, say, gold, for example, gold nanoparticles, for example. So peptides have a surprisingly strong influence on nanoparticle size, morphology, shape, things like this.
Hi,
All right.
this is Mike. I have a couple questions. I can't hosts can't put questions in the Q&A. That's why I have to kind of lurk until everyone was done with the questions. And thank you for all the questions too from the audience. But there's a couple of things that occurred to me. One is that if you were working along with a pixel size that was half of what the pixel size actually was, I would have thought that the defocus that you got, because the CTF, of course, calculation depends on resolution, that the CTF numbers that you got for the defocus would not have been what you had said here. Take the picture at defocus 0.1 micron or whatever defocus you used. It would show up as something quite different, either too much lower or much higher defocus than you had thought because of this pixel size being different. Was that one? All of image processing basically is done automatically, and the only purpose of humans is to apply the sniff test and to detect BS. And the sniff tests can be applied at various levels. Another one of which is when you're doing this rotation, I see that certain directions are stretchy and other directions are much more sharp, and that would of course also say, okay, look at the distribution of the angles that the data was collected, and that should give you a clue. Oh, maybe you have either preferred orientation or you just threw out everything except one orientation. It's not preferred orientation. It's just you selected only one orientation, and that's what you did the reconstruction at. And one of the things that people tells you to do, I mean, that one of the take homes from the CryoSpark workshop, and thanks for that. It was a great workshop, wasn't it?
I'm looking forward to looking back, and I think days 2 and 3 they talk a little bit more about, I would say, that the things that I am going to do next, whereas the first day was like I mean, it did help and inform some of the decisions I made over last week, and I've been chugging away in the background, but I haven't been able to pay too much attention to it working on this presentation. But yeah, it was great. And
what they always do is be able to grab a 3D map and show it in Chimera right away. And that's very important to see. 2D projections or 2D slices through an object really doesn't tell you much about the structure. You really have to, as you see here, rotate it in 3D, but Chimera is a really good tool for that. But okay, those are things that and
I would say the defocus didn't look like, I'll go back now that you said that and do a closer inspection of it, but it wasn't that, it would look pretty normal based on, I would say, Glacios data that I had processed. So that it's kind of strange. Yeah, now that you mention it, that it wasn't something that flagged. I mean, there were definitely moments along the way I was a little bit confused. I was like, huh, that's a little weird. I guess I'll just keep going and see what happens. And I guess your point about preferred orientation, there's definitely some of the views of this protein that are much easier to pick and identify in the micrographs as being real. And so I would say I've trying to be careful in like leaving in a lot of the junk, because if we're looking at this along its long axis, right, it's maybe 70 or 80 angstroms, like from tip to tip. So like that's distinguishable. Looking at this side-on view, like that is a circle that's like 40 angstroms or less. It's like quite small. So yeah, I think I'm trying to recover some of that information. And yeah, because the azimuth, I guess, I don't know the name of the plots, but the orientation plots, yeah, definitely, like there are hot spots. I would say they're not as bad as some that I have seen in my limited experience, but yeah, there are definitely orientations to this that look really nice, and some that look, as you rotate it, not nearly as nice. So
Okay.
yeah, no, I'm looking forward to continuing at this with a little bit more knowledge and more caution. I was sort of at the top of the Dunning-Kruger. I was at the top of what I like to call ignorant hill there in my first workflow, and I've successfully been humbled, and I'm happy to continue learning all this. So
You're
yeah.
really on the right track. That's great.
Yeah, thank you for sharing your learning, and for sharing that it is a learning process, because I think that's not always obvious, but that is the process. And I think we gave you as much work to do. You did a bunch of work to give this talk, and now we've given you a bunch more work after.
Yeah, no, it's good. I told Christina when we were chatting before, my dissertation is due in a week and a half, so and my defense is in less than a month. So this got me off my butt making figures again, really trying to like get towards a structure that I could at least plop in. Yeah, no, this has been great. Yeah, no, I really appreciate the invite out to talk about this. Really excited about our work, the work going on in the lab, and yeah. So thanks again.
All
Yeah.
right. Thank you, Bradley.